Okay, thank you for your patience there. So Ali introduced me there. I work in Newcastle hospitals, lead the Flow Cytometry service here. So in today's talk, I was asked to go over some of the work we do for primary immunodeficiency. So I'm going to look at lymphocyte subsets because I think that's one of the most critical part of that service, really. Include naive T cells in that. And then we've got some other surface stains that we do. And then I'm going to touch on some of the assays we've got that are intracellular stains, and then some of the more functional assays that are maybe inducible antigens or like a functional process. Just sort of for context, in Newcastle, we've got the Great North Children's Hospital, and here we transplant around 30 to 40 patients a year in paediatrics for immunodeficiency, and maybe a little bit more now, actually. And then another 40 to 50 that are related to hemato-oncology diagnostics. And there's also some patients that are transplanted in adult life for PID. So just, I mean, I'm sure a lot of you know all of this already, but just as a sort of intro in terms of what primary immunodeficiency actually is, really the more kind of accepted title in a way now is inborn errors of immunity. And the reason for this is just because they're not always necessarily something that's deficient, as obviously more have been discovered and described and the genetic defect has been identified. There's a lot that actually show a variation in whether it's a gain of function or a haploinsufficiency, and it's not quite as straightforward perhaps as being deficient in a particular cell, but that name is obviously still really commonly used and well understood. So obviously the disorders are all rare, but actually there are hundreds now described, more than 450, and that's increasing really rapidly with the kind of improvements that have been made in their access to genetic testing and what we're able to find out from that. So although they are rare, actually collectively, it's possibly more prevalent than what we maybe appreciated in the past. And really it's essentially some sort of disruption to the immune system. So something's missing or it's present but doesn't work as it should or when it should. And there is some clinical commonality to all of these. There's obviously an increased susceptibility to infection. Autoimmunity is a common feature, autoinflammation, and then bone marrow failure and malignancy often present as the sort of first clinical feature, and allergy, and most have some sort of overlap of these features. If we look at the European Society for Immunodeficiency's website, there's some really really useful information on there about what might give you a clue in terms of a patient who might have a primary immunodeficiency. So these are like their 10 warning signs in terms of what might happen to a child that might make you think that this is more than just a coincidence and that there needs to be further investigation. So it's a kind of a checklist almost. And if you had a patient who kind of fit some of this criteria, it might be that they really need some more thorough investigation into their immune system. And the SCID website's got an equivalent version for adults, which has 6 points on, I think, and then there's the information on there. So it's really useful, and you can kind of see that actually the common sort of feature here is that there's a lot of infections and that they may be difficult to treat and difficult to clear. Even some that would, in a sort of healthy individual, would normally just would clear up quite easily. And then in addition, failure to thrive is quite an important, and a sort of newborn infant that you would need to consider as another feature. And then similarly, you can look at like what type of infection and relate that back to the biology of what would normally sort of be our protection against it. This is a really sort of simplistic version of quite a complex thing, but if the common feature is all bacterial infections that would normally be cleared by antibody, then perhaps there's a defect in antibody production and therefore B cell defect. That would be the sort of train of thought. Similarly, pyogenic infections like Staph aureus, if they were a common feature, it might be that neutrophils are like the cause. So if there is a common theme to the infection, it can hint at what type of cell line you might want to investigate first. Sort of specifically, and for different PIDs, there is diagnostic criteria on the SCID website, and there's others being published. And this, it's interesting to look at. They've got a whole list of sort of well-described PIDs, and they have definitive diagnostic criteria, but they also include some probable, possible, the spectrum of disease, and a differential diagnosis. So it's really, if it doesn't maybe quite fit the textbook definition, if some sort of laboratory test is slightly out of what you might expect, that's a really useful website to sort of consider other diagnoses that might have not been the first thought. I've just highlighted X-linked severe combined immunodeficiency as it's the most common PID that we see, and their definition here includes obviously some laboratory measurements, so less than 10% CD3 positive T cells. Sometimes it's written as less than 300 cells per microliter. It's a different, just a different way of describing it. And then obviously a mutation in the cytokine for common gamma chain, or there's other ways of measuring that, and then the family history. So that would give you a definitive diagnosis. So this is really where you can see the laboratory comes into play. And I think for any patient that is under investigation for a primary immunodeficiency, it's really important to still consider the really common and quick laboratory tests that we'll have available to us. So full blood count and immunoglobulin levels can tell us loads about what might be happening to the patient. So it's really useful that they're done and they're carefully considered. Are they within the reference ranges for that age group? What does the morphology look like? What are the immunoglobulin levels like? It's really important part of the assessment. And similarly, there's clues in terms of what cells may be present and in either in excess or in deficit as to what might be the cause of the patient's clinical symptoms. So that's what you can do really quite quickly. Obviously, from a lymphocyte point of view, essentially the full blood count gives you a total lymphocyte count, but we really need to look a bit closely at the different lymphocytes that we have available and the different functions that they then play in the immune system. And again, even from morphology, they don't always look very different from each other. So it's not, you wouldn't necessarily know without flow cytometry. So the basic lymphocyte subsets that I would imagine is common practice include CD45, which is the pan-leukocyte marker. Lymphocytes are typically bright for CD45, so it helps for gating. And then they're differentiated into T cells, B cells, and NK cells based on the expression of CD3, CD19, and CD16 and 56. The CD3 positive T cells are then further split into CD4 helper T cells and CD8 cytotoxic T cells. And that's a sort of basic lymphocyte subset panel. In Newcastle, we use BD reagents and equipment, and we use the true count method. So I'll just move on the next, our panel gives us percentages and absolute counts. But obviously, if you've got a lymphocyte count from a hematology analyzer, then you can use that and just use the percentages, and it would give you a good indication of the absolute numbers of the different lymphocytes. So in this case, the lymphocytes are gated here in red. They're the brightest for CD45. This is just to get the bead count. The CD3 positive and negative are split, and of the CD3s, they're split into CD4 and 8, and the CD3 negatives are split into the CD19 B cells and 1656 NK cells. And then it gives you a breakdown of the percentages and the absolute counts. And these do vary a bit with age, so it's worth bearing in mind a reference range for the particular age group. And there is some internal QC so that it's past the lymphocytes all add up to greater than 95%, so the lymphosome passes. In a different example, this patient, there are lymphocytes present, and the count would be sort of 1.1 on a full blood count. The lymphosome passes, but clearly there are no CD3 positive T cells at all. In fact, all of the cells look like majority are B cells, and there's very few NK cells. The count is only 44. So really, they're sort of the T neg, NK low or neg really at such low levels. And one thing just to point out, I think if you get a result like this, it depends, I guess, on the sample and the clinical details, what question you've been asked, but always just bear in mind a technical error, because obviously if it's a manual setup, or you could have used the wrong reagent or done a pipetting error or something. So a very laboratory point, but I think it's really useful to remember that you could get a strange result if there'd been an error in the process. It might be worth doing a repeat of this testing just to make sure that it is accurate. In fact, this patient was under investigation for SCID, so it did fit. This is a similar investigation, similar sort of clinical history, but in this instance, the cells were essentially all present and within the reference range. It was a 3-month-old child. So these numbers, some of them look quite high, but I think for a 3-month-old, they essentially were all more or less within a reference range. So initially, from the lymphocyte subsets, there isn't a really significant abnormality there that would raise alarm bells. And this is kind of where I just wanted to go on to mention T cell, just because there is clearly cells present. Actually, then they're not all normal, and they're not the repertoire that necessarily that would be needed in a normal functioning immune system. To actually work that out, we'd looked at naive T cells. So in the top row here, this is a sort of result. So there's CD3 positive T cells. Some are CD4 positive, some are CD4 negative. But of those, both within the CD4 pos and CD4 negative compartment, there are cells that express CD27 and CD45RA. And these are naive T cells. So these are an indication that the marrow is making lymphocytes and that they are going to the thymus and essentially being released as mature naive T cells in a functional thymus, functional marrow. You can use CD45RO. I think a lot of labs do that instead of CD27. That's just the panel that we have in Newcastle. And if you compare that to the patient that I just showed before, there is CD3 cells present. Some of them are CD4 positive, some are CD4 negative. But actually, when we look at the 27 and the 45RA, there's no cells at all in this. And essentially, they've got no naive T cells. And therefore, I think with the clinical picture as well, this essentially did become a question of, is it SCID or a leaky SCID? And in fact, there is some guidance on this. This is the primary Immunodeficiency Treatment Consortium criteria to help differentiate between SCID, leaky SCID, and Omen syndrome. Because in these two, you can get some T cells present, and it could be a sort of false positive, if you like, that the T cells are there. But in fact, there might be things to consider are maternal fetal engraftment, where the maternal cells have crossed up center in the fetal circulation and aren't removed by the fetus immune system because it isn't functioning properly. If that's ruled out, then leaky SCID and Omen syndrome need to be considered. And there is still important sort of cell counts to bear in mind. So the T cells are still reduced in leaky SCID, and in Omen syndrome they can be normal or even high, but they don't function properly, and we can see that partly from the lack of naives. So those cells, I mean, there is some extra criteria here which mentions oligoclonal T cells. I don't have that result for this patient, but there would probably be a very limited repertoire, and they've just peripherally expanded, which is why the total numbers look normal. Most of them are memory T cells. They're not naive, so that fits with this criteria. And in addition, we did do the lymphocyte proliferation on a couple of occasions, and this is just it's not done by Flow in Newcastle, so I haven't gone into it a lot, but these results for the patient are very low, below the 30% sort of cut off for the diagnostic criteria. And actually, when you looked at the clinical details, the patient had a rash, splenomegaly, elevated eosinophil count, and IgE, and they had multiple viral infections. So clinically, and with the test that we had available at this point, it very much fit the criteria for Omen syndrome. Just another point is that we often look at activated T cells, so a measure of how activated and essentially angry the T cells are. And we look at HLA-DR expression. In this case, it was mostly all activated, and that again is an indication of something not right. You might see it in graft-versus-host disease, or maybe just in a really significant infective picture, but it fit with this query around the patient having Omen syndrome. So I just wanted to kind of highlight this in terms of although the lymphocyte subsets typically are excellent, really useful kind of indication of the cellular immunity, it's not always enough. And this just leads us on to mention about TREX and the newborn screening for SCID. TREX are sort of fragments of DNA that are left over from the VDJ rearrangement and the TCR production process. So they're in circulation and relatively easily measured by PCR, and they're a good substitute for naive T cells. They're a good sort of surrogate marker for that, and an indication that the thymus is working. T cells are being produced. So if there's no TREX, it could be a sign that there is a T cell abnormality and potentially SCID. In England, this is currently on trial, so it's not routine practice yet. And if the TREX are absent, then the patients are transferred to a place to get the bloods taken for lymphocyte subsets, and we have to include naive T cells in that process. I know that's quite common practice in some other countries, but it's still on trial here in England. And then another assay that you can do by flow, which is related to SCID, is for the common. So it is you can measure that by flow. In an X-linked SCID, that is common gamma chain deficient. They're essentially absent for that. So it's quite useful. It's very quick, easy sort of screen to confirm what is a suspected diagnosis. Again, from the lymphocyte subset, you'll get a bit of information about your B cells, and if they're absent, really just wanting to point out to remember about B cell depleting therapies. I mean, the rituximab's been the most common one, but there's obviously quite a lot out there, and they're also being used for a broad spectrum of reasons now. So particularly in an adult, remember to check about any treatment options. But assuming that's not the case in the context of investigation for PID, one common cause of a lack of, or the most within a rare disorder, is BTK deficiency. So this assay, you have to permeabilize the cells, but it's quite nice in that essentially when it's normal, this is monocytes expressing BTK, and this is B cells. When it's absent, the patient typically has no B cells or very few, so you can't really measure it because there isn't any. But monocytes also have BTK, but they don't need it to be produced and to reach the circulation. So the monocytes work as a nice surrogate for the fact that the BTK protein is absent. This is like further proving BTK is essential for B cells to develop beyond the pro-B cell stage and reach circulation. So all of these B cells from peripheral blood have to have BTK, whereas in the monocytes, it's not essential for production. So you get a mix split sort of 50-50 in a carrier. And that's quite a nice assay in that it's sort of got an inbuilt internal control. T cells don't express it, so they're negative. And then just further on the B cells are probably more commonly diagnosed in the adult population is CVID, common variable, usually without a known genetic mutation. And these are some sort of defect in B cell maturation means that there's a lack of immunoglobulin. So without the antibody, they're increased infection. They typically have hypogammaglobulinemia and impaired vaccine responses. Some quite clear, definitely low IgG plus or minus IgM, below 2 standard deviations of the normal. So in terms of flow cytometry, the B cell immunophenotyping can be informative for these patients, not necessarily for diagnostics, but maybe for prognostics and treatment options, what might happen. So in this panel below, we've got CD27 and IgD, and that's nicely splits up the B cells between switched, non-switched, and naive. And this is normal, and what you typically expect to see is a mixture in an adult. You can look at CD21 and CD38, CD21 low, and then the CD38 bright, IgM positive transitional. And this is from the Euroclass study, which has been well described and around for a little while now, so you're probably all familiar with it. But those excesses in those groups kind of fit with the clinical picture. So patients have an increase in the CD1 low population will get splenomegaly or granulomatous disease compared to those with the increase in transitional B cells. And they tend to have, will also have low class switch and lymphadenopathy. So it's useful potentially for what signs to look out for the patient's like clinical picture. So that's a lot of the surface stains, and I just mentioned BTK there as well. I haven't mentioned double negative T cells, so these are CD3 positive, 4A and neg, but are TCR alpha beta and are often increased in ALPS. That's another surface stain. And Tregs, so regulatory T cells, are CD4 positive, 25 bright. These, again, you can do this on peripheral blood as a surface stain. And there is an intracellular assay for the protein FOXP3, which, if this is absent, leads to a condition called IPEX. This is useful because it's it's staining procedure, and you can, if there's regulatory T cells present, then FOXP3 will be present. So you don't necessarily have to do that assay if the Tregs are looking normal. So I was just going to go on and mention some of the functional assays that we have. So neutrophil oxidative burst is useful in the diagnosis of CGD. Again, it's quite a straightforward process. Essentially, you encourage neutrophils with the addition of PMA to go through the oxidative burst process. They're stained with DHR, and when they do that oxidation, the enzyme also reduces the stain, which makes it fluoresce. So you go from not fluorescent to fluorescent, and that is a nice example of a normal. Almost all, more than 95% of the neutrophils have shifted right across. And we also do the MBT at the same time. It's essentially the same process, but uses a different dye. And this is, and then you look at the slides down a microscope at the neutrophils. And the top one is without stimulation, the bottom one is with, and the stain has turned blue with the process of oxidation. So they correlate with each other generally really well. And that's a nice example of a normal. One thing to bear in mind is patients with MPO deficiency. So in contrast with MPO deficiency, the oxidative burst will appear absent, but the NBT will be normal. And that's where it's useful to still, I mean, we don't test for MPO deficiency very often. Actually, most patients are clinically well, so it doesn't come up really as a query that often, but it does help if you've got a negative burst assay that doesn't perhaps fit. It might be worth considering testing the MPO expression, because you would get a normal NBT if MPO is present. And that's just because the different stains that I used are reduced by different parts of the procedure. So the H2O2 reduces the DHR not burst assay, and it makes it fluoresce, whereas in the NBT needs to be reduced by myeloperoxidase for it to stain. So without myeloperoxidase, the burst doesn't work, with the NBT does. So that's just one thing to bear in mind if you get a different, if you maybe get some results that are contradictory. And then again with the DHR assay, it's actually it's a really nice post-transplant assay that it shows the sort of proportion of neutrophils that are successfully able to go through that process of oxidative burst. Typically, I mean, if you've got good chimerism, most of the cells, if not all of them, will. If the chimerism starts to drop, then you might see the oxidative burst percentage also drop. In this case, it was down to 20%. And this matches quite nicely with the chimerism, but again, it's quite a straightforward test to do to show the success of the transplant. is absent, there's a few different mutations in the NADPH oxidase that could be the cause. So this is the enzyme in resting state, and then when the cell is activated, they sort of join together and become an active enzyme. So any defect in the GP91, 22, 47, 67, 40, and RAC, they are all have been described as causing a CGD, CGD sort of disease. GP91 is the X-linked and most common, and it's usually the most severe form, so patients probably present younger and more ill, and the burst is entirely absent. Sometimes with the P47 and 67, there's a bit of residual activity. A burst might look reduced rather than absent, or the peak on a histogram might be wider rather than nice. So you can kind of have a clue in terms of the fluorescence as to which part of the NADPH oxidase is affected. And then it is possible to measure these by flow. So there is an antibody to the GP91 P22 cytochrome, and it's expressed on neutrophils and monocytes, and it can be seen on, that's a surface stain that you can test for. Again, that's relatively straightforward. The P47 and 67 are intracellular stains, so it's a little bit of a slightly more complicated process, but it's essentially the same sort of method by flow, in that you get a reduced expression on or absent expression on neutrophils. When we test these, we always run a healthy control alongside, because some of the gating and the shift is quite minimal. But when you compare them to a healthy normal, it's quite clear that if they're deficient in that protein, there's no shift at all. Leukocyte adhesion deficiency is defect in neutrophils being able to sort of tether to and move through the endothelium into the tissues, depending on which protein is defective, depends on which part of that process is impacted. The most common is LAD1, which is autosomal recessive, and it typically presents with delayed umbilical cord separation and a lack of pus, but the neutrophil count is raised. And the neutrophils are essentially stuck in peripheral circulation. They're not able to move into the tissue where they're required. And again, so that's in defect in the integrin beta chain, which is CD18. And again, that can be measured by flow with a surface stain. It's always associated with alpha chain CD11A, 11B, or 11C. So often if the CD18 is absent, those three are either absent or reduced as well. And LAD2 is a lack of CD15, but the process for testing those by flow is all the same. And they just have a histogram peak that moves. In this case, the patient is CD18 absent. But it normally, and again, if you ran a healthy control alongside, the peak would be shifted across to, like, sort of 2 logs across on the histogram. So if we move on to look at CD40 ligand deficiency, this is a type of hyper IgM syndrome. It's termed type 1. It's the most common form of hyper IgM syndrome, and it's due to a loss of function mutation in CD154. It's X-linked. So you get a block of class switching from IgM to any of the other isotypes. And then from a lab point of view, this will typically look like low levels of IgG, IgA, and IgE. There can still be some limited production, so the levels can go into the normal range. That doesn't rule it out, but most commonly the levels will be low. Similarly, IgM is normal or raised. It doesn't have to actually be that high. And the B cell count will be normal, but their full maturation sort of immunophenotype won't be all expressed. It won't be typical. The patients tend to present with upper and lower respiratory tract infections, which is sort of typical of an antibody deficiency, but actually it's not limited to that, and they will go on to have other presentations of viral and fungal infections. Patients often have neutropenia and ulcers and abscesses. So really, it's all of cell-mediated immunity that is compromised. It's not just antibody deficiency. So if we think about the biology behind that, the T cell, which has been activated generally due to the sort of environment around it, the cytokines, once activated will express CD40 ligand. So the T cell and B cell are communicating through the MHC class 2 TCR sort of communication there, and the B cell also expresses CD40 all of the time. So when the CD40 ligand is also expressed and the communication is enhanced by this communication, so the T cells are further activated and the B cells again activated, but then they can proliferate and differentiate and secrete antibodies. And these antibodies at this point might class switch as appropriate to IgG or IgA, for example. So this whole sort of system of class switching really relies on this link between the CD40 ligand and the CD40. So where there's a deficiency, you can see why that doesn't take place and patients become deficient in the other isotypes of immunoglobulin. So from a laboratory point of view, we can test for this. It's quite a nice assay, and it's got quite a good positive control and negative control you can do. So we take normal donor blood, and then the mononuclear cells are separated, and they are labeled there with CD3, so we know we're looking at T cells, and then CD4, so we know we're looking at the T helper cells. These cells haven't been stimulated at all by anything, so they remain negative for CD40 ligand and CD69. When the T cells are stimulated and incubated with PMA, that means they'll express CD69. That's an early marker of activation, expressed really quite quickly by all T cells. So all of the T cells are now positive for CD69. And the T helper cells, CD4 positive, are now positive for CD40 ligand as well after this incubation with the stimulant. So these T's, so you have to, it's quite a lengthy process because of the incubation times and things, but it works quite nicely in activating the T cells, and you can see quite good expression of CD40 ligand. This particular patient, pretty much like the sort of standard presentation in a way, they had recurrent ear infections and presented with pneumocystis around 1 year old. They had a long-standing history of diarrhea. So the immunoglobulin levels were a bit low, and IgM was within the reference range. The initial lymphocyte subsets look normal. So at this age, within the differential would be hypergammaglobulinemia of infancy, which is a transient thing, but obviously depending on the presentation and the severity, frequency of infections, this patient warranted further investigation. The B cell subsets were looked at a bit more closely, and just off this plot alone, with IgD and CD27, there was no memory and no class switch, which you'd expect at this age to have some. It does take a bit longer. It's not always a lot in infancy, but you would expect class switch memory B cells to be present, and there's none. So this is kind of indicative really of CD40 ligand deficiency. When we did this assay, the patient's cells, when stimulated, again, they responded well, and in terms of being able to express CD69, which we were able to detect, but remained completely flat for CD40 ligand. So when we test this, we've got this as a nice positive control. We test a normal healthy donor at the same time, and that one was positive for CD40 ligand. So we know the assays worked, and this, despite successful activation of the cells, there's obviously no CD40 ligand expressed. The patient has since had a transplant, and post-transplant we do the same assay, and now their cells are able to express. So it's a nice indication of, like, a functional indication that the transplant has worked, alongside obviously the clinical presentation and chimerism results. So I mentioned it was one of the type of hyper IgM syndromes. There is others. CD40 ligand defects are the most common and X-linked, and the presentations are similar but have slight variations on a theme. So this table just sums up nicely where you might typically see, say, low immunoglobulins or a bit more variation. The class switch memory B cells being low, or memory B cells, or potentially can be normal. So although this one is the most common, bear in mind there are other more rare defects available that might present clinically in a similar way. And there is the IUIS phenotypic classification, which is a really useful paper for primary immunodeficiency generally. It groups things based on sort of clinical presentation, gives a little summary of what you might commonly find from a lab point of view, as well as clinical presentation, infections. And on here, CD40 ligand deficiency comes under the combined group. So these are immunodeficiencies that affect cellular and humoral immunity, but are generally less profound than SCID. So patients can be a bit older when they are investigated and diagnosed. And there's a little bit of a description there, which is quite useful to think about what clinical outcome might be. And then some lab tests there, IgM, normal or high, or the isotypes are typically low and memory B cells absent. T cells are normal to low. So that's really useful and kind of groups them together in a nice way. And although a lot of these are like incredibly rare, that because their presentations might overlap, this just helps differentiate. It's a really useful paper if you haven't seen it before. Looking at another part of innate immunity is NK cells that are considered part of the innate immune system because of their limited receptor status. But they are like CD8 T cells, are involved in cell killing and antibody production. So they can identify malignant cells or infected cells and destroy them. And the process can be induced in the lab, and certain parts of this can be measured. So it's really important if a patient presents with HLH. I think I've got a slide on next. So it's like a normal process, but just in excess. So these pro-inflammatory cytokines, interferon gamma, TNF alpha, and IL-6 are just produced in a huge excess, and you get this huge inflammatory response. And there's some criteria listed on the right-hand side there as to what would fit with an HLH diagnosis. The underlying cause of that is it might be related to autoimmune disease, malignancy, different forms of treatments like CAR T for malignancy, but there are some inherited primary immunodeficiencies that make patients more susceptible. So we can look at some of these by flow, and if that was the kind of considered, if there wasn't a known cause and you wanted to look at them, there's a few different described genes and functions of those genes, and some of those can be seen by flow. There is a few that affect other sort of processes as well. So in Chediak-Higashi syndrome, for example, you get these giant lysosomes, and you can see those on the blood film. So there might be other indications, but if the typical HLH proteins, they can be assessed. So this is the NK cell killing assay and the or degranulation assay, sorry. And again, the cells are incubated, this time with a cell line called K562, and they're either arrested or they're activated. And the amount of degranulation can be measured by assessing the CD107A expression by flow. So normally this is inside the cell, but as the cells are activated to kill the K562 cell lines, they express CD107. And there's without the this is just in complete medium, without the cells, they're not active. And then when they're incubated with the K562s, they proceed to degranulate, and the cells all express or mostly express CD107A. There's like an indication, if it's this is like at resting, so it might be about 15% would be normal, and then this is activated with the IL-2, which is the sort of promotes the T cells, and this is going to be a significantly higher proportion of the cells that then degranulate. So in certain hereditary conditions, this process isn't possible essentially, and the cells are the granule contents are stuck inside the 107A, which you can kind of just see there. It doesn't reach the surface. And additionally, the perforin deficiency could present in a similar way. So this is part of the granule contents that are released and target the cell for apoptosis by essentially making pores in the cell surface. perforin is like complement component C9, so it's like a bit like the membrane attack complex. And this is just measured, again, it can be measured by flow, so you can do like an intracellular stain and see the expression of perforin in NK cells, which essentially is a nice positive peak there. And a patient with perforin deficiency has no shift in their peak. That's just one point that we do make quite a lot, is that the protein can be present but non-functional, and this is where I guess a limitation of the flow panel, in that it will bind to the protein and fluoresce, so it looks like the expression is normal, but in this case and in some of the other protein expression, we can't necessarily say that it functions in the way that it should. And then one of the last things I just wanted to mention in terms of the role of a flow lab in PID is actually when the patient's been transplanted. So it's a really important way of measuring the sort of success of the transplant and the immune reconstitution. And you can kind of quickly see the NK cells recover, T cells recover, and then you can measure naive T cells and then B cells recover. And it's a really important and useful sort of assessment of a patient's immune recovery after a transplant, and hopefully fixing the initial immunodeficiency at the same time. Those patients that come in via the SCID screening route all get their 6 colour subsets, activated T cells and naive T cells. And so there's a lot, I mean, it depends a lot on the type of transplant, the type of conditioning, and the type of immune deficiency that was diagnosed originally. But there is some good guidance about when you would expect to see immune cells return after transplant. And you can kind of map that process quite nicely. And the NK cells come back first. That's the green line on this graph, followed by T cells. And at the same time that the T cells recover and the new T cells are present, then the HLA-DR expression on them seems to drop, so that become less activated, and the recovery essentially is improving.
Do you think that PID testing should be offered by every flow laboratory or centrally?
Probably a bit of a political question, isn't it? And maybe depends where you are in the world. I think lymphocyte subsets and for me, naive T cells they're quite a straightforward thing, and if you've got a flow cytometer, you could do them. But then, who's interpreting them? Do they have any good experience of PID? Like, is there a good clinical support there for that process? But there's some of the more kind of esoteric assays and the functional, we don't get a lot of requests through the door, so I think they're probably better off centralized, where people can gain experience and maintain a competency in them, because they're quite fragile, some of these cells, and if your processes aren't great, then the assay might not work. And is that because it's genuinely absent, or because we made a mistake? They probably need quite a bit of hands-on experience, so I think they're possibly better centralized.
So another one of the tests where you don't do it often, you don't get the expertise. So
yeah,
people who do most. Yeah, that's great.
It probably, sorry,
sorry, go on.
I was just going to add, it probably fits with what the clinical service is like in your facility as well, because you really kind of need the medical staff to support some of these assays and interpretation of them.
Great, thank you. So, question from Darren North. How do you control such a wide range of tests for such rare diseases?
Yes, I mean, probably with some difficulty, really, especially when you compare to, say, a lot of, I don't know, biochemistry assays that are something that have loads of beautiful controls and calibrators. This is not really that kind of science. You have to understand the cells and know where expression is expected and not expected. And if you've got internal quality control like that, that allows you to know your assays worked, a bit like the CD69 expression in the CD40 ligand assay. So you sort of know your assays worked.
Yeah.
And you test a normal, healthy control at the same time, but that requires someone to be willing to donate their blood So controlling them is difficult, and that I mentioned the travel control, that's really important. And you have to be quite careful that although you don't want to reject samples, but you know, if you've got if you know a sample only works when it's less than 24 hours old and it turns up and it's 48 hours old, I think you have to reject them because you're just not going to get a good answer.
Yeah, totally agree. Can you suggest any EQA scheme for neutrophil function? That's from an anonymous attendee.
I might have to ask you, Alison. So no, there isn't one, is there? But we do a sample exchange with a couple of other centres, and that has worked really nicely. But it's difficult because the more centres you include, the more material that you need, and then you've got to think about the postage of that and how you're going to transport those samples around the UK. So it's not an easy undertaking, but we have done that and it has been successful.
We are in the process of hopefully developing the immune monitoring program to include primary immunodeficiencies and other diseases. So we are going to be sending out a survey. So hopefully we will be able to provide some EQA for that in the not too distant future.
That would be excellent.
Fingers crossed. So a question from Elizabeth Eschel. Do you look at any of the CD11s on, is it LAD1, LAD1?
Yeah, we do. We'll look at them all at the same time. To be honest, we'll just set them all up together because it is quite a straightforward surface stain and stain lights wash procedure. So it's not difficult to, so yeah, we tend to just put them all up at the same time.
Brilliant. Another one from Elizabeth. If you're using CD45RO versus CD45RA, would you only use the bright events?
I'm going to have to pass on that because we don't use RO versus RA, and we used to, but not for quite a long time. So I haven't got a good handle on how that looks in kind of normal naive T cells.
Okay.
Sorry.
It's not your fault. And Berna Yavuk Aksu, I hope I've said your name right. Could you please show again the calculation formula for true count tube? We'll be sharing the slides, won't we? So it'll be on there.
It's on the slides, yeah, and you can have access to them.
Brilliant. Oh, this is a good one from an anonymous attendee. What is the oldest that you've seen a patient present with a PID?
Oh, I don't know. I mean, in adults, definitely. In CVIDs, they can present into middle age. How old's old? I don't know.
Wow.
I can ask the clinical team, the adult clinical team here, for a definite answer.
That'd be good, and we could put it on the Twitter feed. Yeah, that'd be good. So a question now from Katia Irasima Murray. I do apologise if I'm saying your names wrong. Does your lab participate in external quality programs for functional assays? And then what is your experience?
So generally no, outside of the oxidative burst that we're doing as a sample exchange, because I think they're just really difficult to standardize and move around the country, and to have enough blood to sort of send it elsewhere. So EQA schemes are really difficult. That you can't really fix the cells if you then want to make them express an antigen. So I don't think there's too many options around for functional tests. We try and we use either sort of obviously a normal healthy person, then what we understand of the biology of the protein and where it should be expressed, and then how that correlates clinically, and then actually ultimately with a genetic diagnosis. If we think something's CD40 ligand deficient and then that's confirmed with genetics, it kind of helps validate that assay, but that's not a quick fix. Obviously, it can take a little bit of time.
Thank you. Oh, sorry, excuse me. So I've got a question here from somebody anonymous. Thank you for the excellent talk. Do you think there is a place or clinical need for the PIDOT and extended classification tubes developed by Euroflow with associated normal range databases?
Yeah, I think they're excellent, to be honest. I mean, they're difficult to introduce perhaps to NHS labs. Maybe financially it's not the easiest, but they do have a really good panel on there, and obviously the fact that that's standardized across Europe would give you a lot of confidence in the clone choice and the fluorochrome choice. It would, yeah, that would be a really nice way of kind of getting that standardization and getting the experience, because you could share that data a lot more readily.
Yeah.
So I do think they're good. They look excellent, but it's just realistically of putting how to get those into NHS facilities difficult.
So another anonymous one, just looking at time. Yes, we're fine for a few more. What other stimulants other than PMA can we use for DHR assay?
Good question. I'm not sure. We've never really tested anything else other than with PMA. I can certainly look into it. I know there used to be a kit available that isn't anymore. That might have used something else. Some people use E. coli as another method alongside PMA. They'd sort of run the two together, though, as I understand it, and compare. But yeah, I could look into that, but I'm not sure.
Thank you. One from Athalia Yanaki. Does the TRBC antibody sorry, hang on, I've got to reread this again. Does the TRBC antibody offer anything in PI diagnosis as it can in T-cell leukemias?
Good question. And so we do use the TRBC1 antibody in our T-cell leukemia diagnostic panel. And we have talked about this before, but I don't have any working experience of it. So we do the TCR-V beta assay, which looks at the variety of TCRs that the T cells express. And obviously, if they're very restricted, like an oligoclonal picture, it might be an indication of omens or maternal fetal engraftment in a PID scenario. And I wonder if you looked at those T cells using the TRBC1, whether they'd all be clonally restricted, and that antigen would then all be positive or negative. That would be my working theory, but I haven't actually done that test, so I don't know. I don't know if that is published anywhere.
Okay, thank you. Another one from an anonymous attendee. Have you had many cases using true count TBNK assay when T cells are present but appear to be negative for service CD3 expression and require washing to demonstrate service CD3 pos cells, e.g. in case of cold agglutinins?
We haven't had many cases, no, but that has occurred. And I think so we don't usually pre-wash our samples, like that's not standard practice, but there are some patients that we seem to have to do it every time. So it must be something in the serum, whether it's medication or paraprotein, or I'm not really sure. But some patients do have that, and so it's always in the back of our minds that a pre-wash could be really useful. And if we got something that was really unexpected, but like when I said repeat the assay in case you've used the wrong reagent or something, a pre-wash is another sort of go-to. But we don't see it very often with CD3, to be fair. It doesn't disappear and appear negative when that hasn't happened very often.
Brilliant, thank you. So I think we have time for one last question, and I think this is a very topical one. With IVDR requirements, have you looked at Neutrophil burst kit XBio, and how did you find it?
No, we haven't. But yeah, the IVDR requirements question, yeah, it is a big topic at the minute, and it does raise some concerns around some of our slightly more esoteric assays, shall I say. No, unfortunately, I haven't looked at it, but it's something that we might have to consider moving forward.